Sawasdee We are from Panatia High School
to presenting the study on food odor deodorant spray from tannin extract.
We chose to conduct this project because first is food odor, which is a primary reason to study this topic. Second is sweat stimulation. When bacteria interact with sweat, they produce unpleasant smells. Additionally, if bacteria enter the body, they can affect the immune system. Another important reason is the impact of synthetic chemicals such as climate change, water pollution, air pollution, and negative effects on human health. The plants used for extraction include guava leaf, cassava leaf, and bamboo leaf. The research focused on developing leaf extract spray for food odor. The first experiment aimed to study S. aureus and E. coli inhibition of tannin extracted from guava leaves, cassava leaf, and bamboo leaves. Inoculate S. aureus and E. coli into nutrient board straight on page Cow-Agar. The diffusion method was conducted. The diameter of inhibition zone was measured by using a vernier caliper. These are the results of the diffusion method on S. aureus. It can be observed that this extract produces a different inhibition zone size. Similarly, the discs were placed on E. coli in the same, and the inhibition zone size also vary. The extract obtained using 75% ethanol exhibit a larger inhibition zone than those extract with 95% ethanol. When ranking the antibacterial effectiveness against S. aureus and E. coli, guava leaves extract show the largest inhibition zone, followed by cassava leaf extract, while bamboo leaf extract had the smallest inhibition zone.
The second experiment aims to investigate the amount of tannin extract obtained from guava leaf by Folin cell culture method. The graph represents the absorbance value of standard tannic acid and the dilute guava leaf. While using the absorbance value to calculate the tannin content in the extract, at value concentration, the tannin amount were determined as follows. The third experiment aims to determine the minimum tannin concentration required to inhibit E. coli and S. aureus. The method follows the same procedure as in experiment 1, but instead of using the different plant extract, guava leaf extract was applied of concentration of 10, 20, 30, 14, and 50 microliters. From the diffusion method, it was observed that only 50 microliter concentration produced an inhibition zone. Based on the inhibition zone size, the extract was more effective in inhibiting S. aureus than E. coli. The fourth experiment aims to develop a food deodorant spray using tannin extract. The method followed the same procedure as in experiment 1, but instead of applying plant extract, different spray formulation were tested. Formula 1 contained antibacterial ingredients that have been tested for effectiveness. Formula 2 without antibacterial ingredients. Formula 3 contained 50 microliter of tannin extract. Formula 4 contained undiluted tannin extract. From the diffusion method, it was observed that only Formula 2 did not produce an inhibition zone. Based on the inhibition zone size, Formula 4 has larger inhibition zone, followed by Formula 3 and lastly Formula 1.
Experiment 5 testing the effectiveness of the deodorant spray in inhibiting bacteria from feet and salts. The procedure for this experiment is the same as in experiment 4, but the bacteria used to test the spray effectiveness were collected from feet and salts. From the distribution method, the results appear similar to those in experiment 4. Based on the inhibition zone size, Formula 4 had the largest inhibition zone, followed by Formula 3 and lastly Formula 1. Tannin can inhibit proteins and enzymes and is more effective against S. aureus than E. coli. Due to different intestinal wall structures, the inhibition occurs at a tannin concentration of 4.36 grams, but bacterial inhibition increases as the tannin concentration increases. This research project aligns with SDGs Goal 13, 14, and 15, which focus on environmental conservation and the sustainable use of natural resources, utilizing tannin extracts as a substitute for synthetic chemicals helps reduce environmental impact in several ways. It lowers greenhouse gas emissions, protects marine resources from chemical contamination in water sources, and promotes the sustainability of terrestrial ecosystems. The suggestion is test the food deodorant spray made from tannin extract or individuals who trust the product and willingly consent to its use submit the spray for safety testing on the skin by the Department of Medical Science. Explore the application of tannin extract for water beneficial uses. The example of applications are in the agricultural industry. Tannins can be used to reduce the use of antibiotics. In the latter industry, tannins serve as a natural tanning agent replacing synthetic chemicals. In the pharmaceutical industry, tannins act as antimicrobial compounds in wound healing treatment and help combat bacterial infections.
Thank you for listening.